Good morning. Our lesson today is based on the steps of insulin production. There are 5 steps involved. The first step is to obtain a copy of the gene from a healthy human being. From the beta cells of the islet of Langerhans of his pancreas, we remove the mRNA. This is the mRNA which we have removed. This mRNA codes for making insulin. Now, because this is an mRNA, we have to convert it into a DNA. We have to insert a DNA into the bacteria. So when we're converting it into a DNA, we use the help of an enzyme called reverse transcriptase. You have already learned the use of this. This reverse transcriptase can convert an mRNA into a single strand of DNA. This DNA is now called as cDNA, a complementary DNA. The mRNA is then removed. We don't need the mRNA as such. Now we have a single strand of the DNA. We're going to work on this single strand of DNA. What we do is we take this DNA as a template and we make the second strand complementary to it by taking the help of an enzyme called DNA polymerase. Now this DNA polymerase, as it moves along the single stranded DNA, it makes a second DNA, and you can see these two are complementary and we have 2 strands of DNA, and this part of the DNA, this is the gene that codes for making insulin. Now we have only a single strand of DNA. We need single piece of DNA, single molecule of DNA. We need to make many copies of it. Then only we can manufacture insulin. For that, we do a process called PCR. It's called polymerase chain reaction, where we use a series of steps. When a single molecule of DNA, double-stranded DNA, by the end of this cycle will be converted into 2 copies of double-stranded DNA. One parent is used and we're making 2 copies. The process related to this